Okay, I think it is visible now.
Yes, sir.
Okay, then shall we start?
Yes, sir.
Okay, very welcome to this elective course. And the name of elective course that you should be all aware is that of biology of disease, right? Uh, First, let me tell you what is the basic objective of this course, right? Well, we have started this course for you to understand the very basic biology behind the some of the very common diseases, right? And the objective is not like to mug up all the life cycles and metabolic pathways and reactions that I'm going to eventually tell you. The objective of doing this course is like to have you a basic understanding of how these diseases are caused, and most importantly how people are trying to manage these diseases. Okay, how whatever knowledge that we have accumulated so far in the past few decades of research, how it can be utilized to find out new therapeutic strategies, or how things can be done using existing bioinformatics tool in case of our bioinformatics course, so that it would give you a power to think ki, okay, this is the disease and this is what we know, and this is how bioinformatics as such can be implemented to finding out some therapeutic options, or to design some therapeutic strategies that can be used for developing either new vaccines or either new drug molecules as such. Okay, so yes, am I audible? Is it okay?
Yes, sir.
Yes,
Yes,
yes.
sir.
Okay, please let me know if there's something goes wrong. Okay, okay. So
Sir,
this course, yes.
sir. Niha send a message. Niha Beragi that let her to the group, sir. She message.
What?
Niha Beragi message in the group. Ask you to let her in group. In this
Okay, she wants to let in, right? Just
group.
a minute, huh?
Sir.
Just a minute, just a minute.
Yeah, yes, sir. Yeah.
Okay, is there anyone? Because I'm recording this thing and everything is being recorded now.
Yeah, she joined, sir, actually. I showed on
Okay,
the
now we have
prompt.
21 students, right?
Yeah.
Okay, is it visible now?
Yes, sir.
The presentation?
Oh, yes, sir.
Yes,
Okay.
sir.
Huh. Uh, well, so this course, it has been modularized in each and every context. Like first, we have two kind of diseases to deal with. Most of the time we would be dealing with these diseases. First kind of disease is something called as communicable disease, and the next division of disease would be non-communicable diseases. Okay, so communicable diseases are those which are, you know, transmitted by some bacteria or parasite or virus, right? And non-communicable diseases are some diseases that are not caused by external pathogens as such. Okay, so these might include like cancer, diabetes, arthritis. Okay, so these diseases are not caused by the external parasite, okay, or external pathogenic organism as such. Okay, so the very first section of our syllabus would be about pathogenic diseases, right? And in these pathogenic diseases we have got three categories. First, we would deal with some parasitic diseases. In parasitic diseases, we would be talking about life cycle of plasmodium, and what are the therapeutic opportunities that we have developed so far for controlling malaria that is caused by this plasmodium. right? After Plasmodium, we would be talking about another parasite called as Leishmania. It causes the disease called as Leishmaniasis, right? Then we would talk about the basic concept of the life cycles of those pathogenic Leishmaniasis organism, and of course we would talk about the therapeutics, what are the drugs and what are the therapeutic strategies and how those drugs act and function. Okay, then after that we would talk about trypanosoma. In the same way, first we would learn about its biology in terms of their life cycle and everything, and then we would for each and every disease we would talk about what are the therapeutic options that are available. So in this way you would be kind of in a position to know, okay, this is the biology that we have known about a certain disease, and using this information, this is how people have devised strategies to control that disease. That is what the main objective of our paper is, right? Then when we cover up with these parasitic diseases, we would move on to bacterial diseases. Okay, for example, we would be learning about that Mycobacterium tuberculosis in more detail as a model bacterial disease. Okay, we would also deal with viral diseases, okay, like maybe HIV. Okay, uh, I would try to incorporate our SARS coronavirus COVID-19 disease as well for this year. Okay, then once we finish it off with this communicable diseases, we would deal with two major non-communicable diseases like cancer and diabetes. Okay, so I hope you guys get what will you be studying throughout this semester now, right? Is it
Yes,
clear?
sir.
Okay.
Yes, sir.
Okay. Now, well, the very first disease that I have selected to discuss is a parasitic disease caused by an organism called as Plasmodium, right? And you guys might be aware that Plasmodium causes a disease called as malaria. Okay, तो malaria कैसे होता है? It is caused by Plasmodium.
That's what.
Okay,
Yeah.
so let us get introduced with this concept. So when science was not very much kind of advanced, well, previously it was thought that this malaria is caused by air of swamp and marshy land. Okay, so in Italian, malaria stands for mal, that is bad, and area, that is air. The disease that is caused by bad air is called as malaria in Italian. Okay, so that was quite long time ago, but then in 1847, Mickel first observed that these disease can be caused due to infection of some parasite. Okay, then furthermore in 19th century, there was a physician called as Charles Leveran. What he demonstrated that this disease, the causative organism of this disease is present in blood, and it do not caused by polluted air. Okay, and later on in 1907 he worked out, or you can say he got Nobel Prize in Physiology and Medicine for working out the details of life cycle of this parasite. Okay, so just to let you know that this disease has got a lot of historic prevalence. Okay, so well, this disease has caused a lot of trouble in military history. Okay, specifically during Second World War. Okay, and it is said that this disease in Vietnam, that malaria killed more combatant during that Vietnam War than bullets. Okay, and this was not the only time when some biological agents like malaria has uh, has controlled the history that we have known so far. Well, so I would also like to cite another example Do you know that uh, the epidemic that we are facing right now, okay, the COVID-19, similar epidemic has been observed some 100 years ago in 1918. Okay, do you know what that epidemic was?
Spanish flu.
Yes, that was Spanish flu. Okay, Spanish flu was something that has spread the COVID has spread right now. And
Mhm.
one of the major reason for spread of that Spanish flu was attributed to First World War. Okay, because during 1918 it was outbreak of First World War, and when the troops were sent in different parts of world, there went your influenza virus. And in
Yeah.
this way, these diseases, this pathogenic organism has always had a very big role in shaping the history that we now know. Okay, but that is just in a kind of historical account. Well, let us discuss with habit and habitat of our malaria. Okay, or specifically we can say what is the habitat of this disease, or this sorry this plasmodium. Okay, so if you consider, as you can see over here on this slide, uh, this pathogen, plasmodium, is an intracellular pathogenic blood parasite. Okay, intracellular means it passes from one cell to another cell, and it is a blood parasite, right? It spreads through blood. Now it is not like this disease is confined to specific region, but it is cosmopolitan in distribution. As you can see in this map, this disease is widely spread in tropical and subtropical region. So now, what are tropical region? Tropical regions have warmer climate. Okay, and the region which are somehow present around the equator, well, those are included in tropical region. Okay, so whatever has been shown in this red color, this is called as tropical belt. Okay, and of course, to the north and to the south of the tropical belt, we said that we have subtropical region. Okay, so now the red region indicates there is the highest prevalence of this parasite in tropical and a bit of subtropical region. So can anyone give me an idea, or can anyone well, I am open for discussion ki why these tropical and subtropical region might have highest density of infections than the more colder regions.
Um, Maybe, sir, the higher humidity and the higher temperature, it is more sustainable for the parasite to survive and propagate compared to some drier regions.
Okay, okay, okay. You are in the right direction of thinking.
Yeah.
But there is also one factor that I would like to discuss before we answer this question. Okay.
Okay.
The factor being that this is a vector borne disease. That means it
Mhm.
requires some arthropod mosquito to
Mm.
transfer this parasite from one host to another host. Okay,
Yes.
and in case of malaria, that vector we term it as anopheles. So anopheles
Yeah.
mosquito is responsible for spreading the disease from one host to another host. Okay,
Yes.
so now this anopheles mosquito can survive at 15 degrees centigrade. Okay, even
Mm.
if we move on away from the temperate region and towards the north pole or south pole where the temperature decreases, it can somehow survive till 15 degrees centigrade. Okay, but your plasmodium is not capable of completing its life cycle at such a low temperature.
Okay.
Okay, although the vector can survive, but for completion of its life cycle, plasmodium cannot bear that cold temperature, and therefore most of the infection is found to be populated in warmer part of the world as compared to the colder region.
Okay.
Okay, so this is the basic reason of the highest prevalence of this disease in most of the tropical and subtropical region rather than the colder region in Russia and most of the Europe. Okay, it is not like
So what
that.
will you say is the critical temperature?
For what?
So what will be the critical temperature to maintain the life cycle properly then? Like
Plasmodium
if
requires
since
warmer temperature. Okay, well, it is around 35 or 35 or 40 degrees. It is most active in those temperatures, and that
okay.
temperature is usually maintained throughout the year in tropical regions,
Mm-hmm. Yeah.
right?
All right.
Okay, now let us
Yeah.
talk about host of this disease. Okay, so okay, now it is said that this plasmodium has two hosts, okay, as we have discussed so far, and the organism that require two hosts are called as digenic. Okay, to complete their life cycle, if it requires two hosts, we said that the organism is digenic in nature. Like, okay, do you get it? Like Plasmodium requires one host, female Anopheles mosquito, and another host, man, to complete its life cycle. Therefore, we say that it is a digenic organism. Okay.
Yeah.
So now, what might be the advantage of selecting two hosts for continuation of the life cycle? Can anyone just have a guess? Okay. First, being that if one of the host dies, then of course the parasite would survive because it has got another host to continue its life cycle, right? Is it okay? Okay.
Mhm.
So
Yes, sir.
the thing is ki
Yes.
means it gives an evolutionary advantage to this organism ki even though if a larger number of population of any one of the host is deprived by certain geographic or say some disease event, the species would still survive and continue to flourish further. Okay,
But
so
sir, I have one question. So
yes.
if there are two hosts, which
Mhm.
is absolutely important to
Mhm.
complete the life cycle.
Mhm.
So if by somehow access to one of the hosts is disturbed or is stopped, then
Mhm.
wouldn't the life cycle be suspended in a it wouldn't be it be in a suspended state because एक host कम है तो फिर we cannot complete the life cycle.
Yes, but that
Uh,
is I will come to that thing. Okay, so
all right.
it is not like कि humans are the only host for your parasite. Okay, ये बाकी के host
Okay.
में भी होता है, बाकी के mammals में भी
Okay.
होता है,
Okay. Okay. Yeah.
right? ठीक है. So let us
Okay.
continue with our discussion कि इसका जो life cycle है, well, that is quite very complicated, and it has got two phases. Okay, so the asexual phase of the life cycle of this parasite, what we call it as endogenous phase, that happens in man. Okay, and the next phase, that is called as sexual phase or exogenous phase, that occurs in mosquito and specifically in female Anopheles. Okay, so in this way, man is called as primary or definitive or final host, and female Anopheles is called as secondary or intermediate host. Okay, and this female Anopheles is acting as a vector that would transmit our parasite from one human to another human. Okay, so I should also tell you that these male mosquitoes are not considered as vector, as they do not have any role in transmission of parasitic disease since they do not feed on blood. Okay, now female Anopheles need to feed on blood. She need to take that blood meal in order to complete her reproductive cycle. Okay, but that is not the condition for male Anopheles mosquito. Okay, so that is why only female Anopheles mosquito is responsible for transmission of this disease. Okay. Okay, so now let us talk about what are different species of Plasmodium that exist. Okay, so well, say for example, there are numerous species of Plasmodium that infect reptiles, that infect birds, even man and another mammal. Okay, so there are 4 species of Plasmodium. I am counting on very 4 popular species, okay, which cause malaria in man. Okay, all these species are very specific regarding their primary host. Okay, so well, say for example, Plasmodium ovale, this species is very rare and it has been reported from West Africa, South Africa, Russia, Palestine and New Guinea. Okay, while the other 3 species, say for example your Plasmodium vivax, Plasmodium malaria and Plasmodium falciparum, well, these species are found out almost everywhere. Okay,
Excuse me, sir.
yes, yes.
Did you change the earlier slide? Like, because I can see only the host slide currently on the screen, so that's why I'm
Okay,
asking.
yes, yes, yes.
Yeah,
Oh yeah, now it's there. Yeah.
yeah, now it's
Okay,
there.
I
Yeah.
usually get carried out by this thing. Okay, so as you can see,
Yeah.
there are 4 species, okay, that
Mm.
infect human. Plasmodium ovale. So this Plasmodium ovale is very rare and it is found in specific region like West Africa, South Africa, Russia, Palestine and New Guinea. While the remaining 3, Plasmodium vivax, Plasmodium malaria and Plasmodium falciparum, those are very common and you can find it on different location. Okay, so now let us talk about the structure of our Plasmodium, or what we call it as ultra structure of our Plasmodium. Okay, uh, just to know, I would like to let you know that this Plasmodium demonstrate different morphological stages throughout its life cycle. Okay, so it would go on changing its morphological state when it transfer from mosquito to human. Okay, it would have some different morphological state when it initially infect into liver. It would take different morphological state when it would go into RBCs. Okay, we are going to talk about that in a bit later. Okay, but the most popularly taught structure of Plasmodium is that of a state called as sporozoid stage. Okay, so are you comfortable with this concept that it can have different morphological state, and one of the morphological state is called as sporozoid state. Okay, the figure that you see
Yes.
is a typical sporozoid that is very infective stage of Plasmodium. In this stage or in this morphological state, it will infect human being. Okay, so as you can see, they are small sickle shape and fusiform spores. Okay, fusiform means the ones that tapers on both the end. Okay, and these are highly motile organism that tapers at both ends and they measure about 14 micrometer long. Okay, they are 14 micrometer long and about 1 micrometer broad. Okay, is it okay? Okay.
Yes,
uh,
sir.
okay,
Yes,
and
sir.
as you can see,
Yes, sir.
these parasites are they are surrounded by an outer firm and elastic layer called as pellicle. Okay, and there are microtubules which we are going to deal with in more details that help in the wriggling movement of its body. Okay, so they move in a wriggling motion. Okay, so this is some this picture has got some more features of this parasite. Okay, uh, now this picture I have taken it from the paper that I have mentioned it over here. Can everyone see this? This paper, Gracia et al. Okay,
Yes, sir.
so from this paper I have
Yes, sir.
taken this particular figure. So these malarial parasites per se, well, they belong to a specific class of parasite called as Epicomplexan parasite. Okay, so these Epicomplexan parasites, well, they have evolved some special organelles that are collectively called as the epicomplex. Okay, so what do I mean to say from this is ki all the parasite which has got this epicomplex are called as epicomplexan parasite. Okay, so malaria is included in this epicomplexan parasite. Okay, is it okay? ठीक है. So
Yes, sir.
now,
Yes,
well,
sir.
let us
Yes, sir.
study in more detail what exactly this epicomplex is. So first thing is, I hope is very clear that the parasitic organism having this epicomplex are called as epicomplexan parasite. Okay, so this epicomplex is formed by four different structures, or you can say four different special organelles. What we term them as rope trees, micronemes, the apical polar ring, and conoids. All of these four special organelles collectively form something called as the epicomplex. Okay, so of these four special organelles that these parasites have evolved, of these four, two are said to be secretor organelles. That means these rope trees and micronemes, which are considered as secretory organelles, they are responsible for producing certain chemicals or certain product that enables this parasite its mobility, its addition to the cell surface during the infection. It helps in host cell invasion, and these proteins or the product that are secreted by rope trees and micronemes, they are responsible for the formation of something called as parasitiforous vacuole. Okay, so now let me tell you something about parasitiforous vacuole first. Okay, what happens is that when our parasite goes and infect human cell, okay, it just do not go around wandering inside the cell. Okay, like it is free to roam anywhere. Okay, so instead what it do is that it creates a separate isolated environment for itself, which is membrane bound, and whatever development that happens, so far this parasite is concerned, that happens in that parasitiforous vacuole. Okay, is it clear? Shall I
Yes,
move
sir.
forward? Okay, so this is what is all about our parasitiforous vacuole. Okay, the next component of this epicomplex is something called as the apical polar ring. Okay, as you can see over here, this is present on the extreme apical end of the parasite. Okay, this portion is called as apical polar ring. Okay, so basically this is nothing but a tight knit tubulin base structure. Okay, and it is said that it is evolutionarily derived from ancestral flagella components. Okay, now one of the theory that explains or that tries to demonstrate the evolution of this parasite, it is said that the ancestor of these plasmodial parasites, well, they had flagella, and in due course of evolution, then they might have got rid of that flagella, but still there are remnants of these flagellar components in form of a polar ring. Okay, So along with this, the next typical structure that is present in our parasite is something called as conoids. Okay, so these conoids, well, these are supposed to be present in the lumen of apical polar ring. Okay, and the core of these conoids is present of some tubulin polymer. Okay, so the exact function of these conoids is not very clear, but there are few papers that are published that has linked the conoids with secretory function, or it might be involved in its motility, or even in chemosensory functionality. Okay, so these four organelles, spatial organelles, collectively are called as what? They are called as Epicomplex. Okay, is it clear?
Hmm. Yes, sir.
Okay, please let me know if I am going fast. Okay, you are very I'm kind of very free to get in conversation with you guys, right? It should not happen that I go on shouting and you get lost with what am I dealing with. Okay, please let me know if I am going fast. Okay, good. ठीक है. So
Okay.
now uh, let us talk about certain proteins that are associated with these organism or these specific organelles. Okay, well, as you can see over here in these micronemes and the surface of this parasite, there are certain proteins that are associated, like trap, like uh, aspect, like mabel, like ppl 125. Well, these are all proteins that are specifically associated with this micronemes and surface of this parasite. Okay, while this CS protein, SALSA, STRAP, LSA3, SPATR, PXSR, PFEMP3, well, these are specifically located on surface of these parasites. Okay, and there are certain proteins like LSA1, like LSA3 and strap that constitute the major factors, or that constitute the major player in maintaining the parasitivorous vacuole. Okay, so why I am telling you about this protein is that somehow these proteins are very typically present in malarial specific organelles. Okay, in parasitivorous vacuole where development of this parasite happens, as well as on surface of this protein. So this might eventually help you in thinking in a such a way कि, okay, if I study the physiology of these proteins in more details, won't they act as a good therapeutic opportunity or therapeutic drug targets, right? Okay, so I now would like that you people, well, I have given you the paper already over here. So you people, what you are supposed to do for today's assignment is that you are supposed to read this paper, which gives details of all these special proteins which are present in the plasmodial parasite. Okay, and you are supposed to write a kind of write up. Okay, in which you describe ki what these proteins are and what is its function in physiology as well as pathogenesis of the plasmodium as such. Okay, do you get what the
Yes, sir.
assignment is?
Yes, sir.
Okay,
Yeah.
sir.
Okay, you have
Yeah.
this paper, you need to download it. It is freely accessible in PubMed Central, and what you are supposed to
Okay.
do is read it. It is a review article, and it gives details of all these proteins. Okay,
Okay.
so
Yeah.
this would enable you to think in a way कि okay, these are the proteins that can be targeted even in drug target, and since these are proteins which are present on surface of the protein, maybe we can use it as an antigen and create some peptide based vaccines, subunit vaccines we can say. Okay, but for that you should be able to know ki when these proteins are expressed, what role do they play in pathogen physiology and everything.
Yeah.
Okay, so
Yes.
I advise you to read this paper. Okay, read about all these proteins, make a write up, and then submit it me on my email, or maybe I would submit it on maybe you can submit it on that Moodle platform.
So
Okay.
Okay.
did you share the paper with us somewhere, or like we will look this up on Google ourselves?
You can just take a screenshot of this, and
Ah, okay.
maybe you can search it out. Okay, and of course if you wish I can share it on our WhatsApp group.
Ah, okay, Ashok.
Okay,
So I found it. Maybe I can type it in the chat for everyone if they're
okay,
interested.
okay, okay. ठीक है. Okay, so that was all about our epicomplex, right? Well, there is one another very unique structure that is present in this parasite. What we called it as a picoplast. Okay, and this is very essential organelles for parasite survival. It is very similar to chloroplast that is specifically present in plants. A picoplast is specifically present in uh, apicomplex sun organisms. Okay, so besides these unique structures, तो कौन से structures अपन ने पढ़े हैं अभी? We have learnt about epicoplast. Okay, we have learned about this epicomplex, which is formed of specific organelles like rope trees, micronemes, apical polar ring, and conoids. Okay, is it clear?
Yes. Yes.
Okay, तो इनके अलावा well, there are several cytoskeletal elements that are involved. Well, say for example, like actin, myosin, a bundle of microtubules, of course, and networks of filament like protein. And using all these proteins, well, this sporozoid somehow is able to get mobilized from one location to another location in the host. Okay, so I hope I have cleared a lot more things on the structure of this parasite. Okay, so I think now is the time that you people start out asking questions if there are, or shall I proceed further?
Sir,
Yes.
sir, I want to ask assignment. Which proteins should we clear according to
Use
this
all these
paper?
proteins and write a paragraph maybe.
Okay, no, about all these proteins of this parasite,
I will let you know in
Plasmodium
that
falciparum.
WhatsApp group, or maybe I will send an email. Okay, is it okay?
Okay, sir. Okay,
Okay,
sir. Thank
so
you.
I have only
Yes, sir.
discussed about the specific structure that are unique to this parasite. Okay, so along this unique structure, of course, since it is a
Okay.
eukaryote, it would be having nucleus, right? Nucleus would be associated with endoplasmic reticulum. Okay, there would be Golgi complexes for secretory
Yes.
purposes. Okay, do you get this? Of course, there would be mitochondria for energy production.
Yes,
Okay,
sir.
all the other features of eukaryotic cells would also be present in our parasite, but these are the special features that these epicomplex and parasites have. Okay, good. So now
Okay, sir.
let us begin with the life cycle of Plasmodium vivax. Okay. ठीक है. So
Yes,
for,
sir.
yes, is there any specific question? No, shall I proceed?
Yes, sir.
आगे
Yes,
बढ़ो.
sir. Yes, sir.
Okay, Please let me know if you guys are uncomfortable or you didn't understand anything. Huh? Please let me know. Okay. Now, the life cycle of this protein, life cycle of this organism, it involves three major phases, as you can see. Uh, the most dominant phase, and that is related to human, we would call it as schizogony. Okay, and this schizogony usually deals with the asexual multiplication. Okay, after schizogony, there is another phase in the life cycle of this pathogenic organism. We call it as gametogony. Okay, so this gametogony begins in the primary host, that is called as man, but it is somehow in man it is unable to complete this gametogony, and it has to change its host so that it can complete this phase in another host, or what we call it as secondary host. So all in all, what I want to say is the next phase, that is gametogony, it begins in primary host but it ends in secondary host. Okay, and in secondary host there is another phase called as sporogony. This sporogony is related to asexual multiplication in secondary host, that is female Anopheles. Okay, so for the sake of simplicity, we would be considering the events that happen in the life cycle of Plasmodium vivax. Okay, wherever it is relevant, I would let you know that how things happen differently in Plasmodium ovale, or how things happen differently in Plasmodium falciparum. Okay, so our model organism to study the life cycle of Plasmodium would be Plasmodium vivax. Okay, good. So what do you think might be the very first stage or first step of the parasite? That would be inoculation, right? The
Yes.
parasite first must get inoculated into human body.
Yes, sir.
Okay, so well, let us first talk about the inoculation first, right? Inoculation means getting the entry of the parasite into human body is called as inoculation.
Mm.
Okay, so typically the parasite in the form of sporozoite are inoculated in the blood of healthy human being. Okay, when an infected female Anopheles sucks its blood. Okay, तो ये कब घुसते हैं अपने शरीर के अंदर में? When an infected Anopheles mosquito that is infected by this parasite goes and bites to that human being. Okay, so now the mouth part of female Anopheles are specially adapted for piercing and sucking. Okay, the saliva of the Anopheles mosquito contains some of the biochemical elements which prevent clotting of the blood while sucking. Okay, as we know that the male Anopheles mosquito sucks the nectar from flower, it do not have any role to play with inoculation as such. Okay, so now, well, let us consider that the mosquito, female Anopheles mosquito, has sit on that human being, and it has been already infected with our parasite, and after biting, now thousands of sporozoid would enter into the bloodstream of the organism, that is our primary host, that is human being. Okay, so now after the parasite has been inoculate in sporozoid stage. Okay, जब inoculation होता है blood में तो क्या होता है? They are in sporozoid form that we have discussed so far. Okay, so after about half an hour or after a complete hour of inoculation, the parasite don't remain in blood for a longer time. and they begin to leave the bloodstream and finally get into the parenchymatous cells of liver. तो सबसे पहला टारगेट क्या होगा आपका पैरासाइट का? टू गेट रीड of the blood vessel and go to some safer place. Okay, तो जैसे ही मॉस्किटो इनोकुलेट करेगा तो द पैरासाइट हैज अ डिसएडवांटेज ऑफ बीइंग इन ब्लड सर्कुलेशन. कौन से डिसएडवांटेज रह सकते हैं? देयर आर इम्यून सेल्स सर्कुलेटिंग क्वाइट अराउंड, बरोबर है?
Yes, sir.
And if these immune cells find out कि okay there is parasite, and eventually an immune response would be woken up, and the immune cells would clear out the parasite before the final infection. Okay, so the first major thing that this parasite need to do is to get out of blood circulation and get to certain very safe place. And this parasite has choose what organ from human body? The liver cells. Okay, तो इसका पहला ठिकाना कौन सा होगा? Liver. Okay, so sporozoite now enters into liver sinusoids, and then they get entry into liver's parenchyma, or what we call it as the hepatocytes. Okay, बात समझ में आ रही? Is it okay?
Yes.
Okay.
Yes, sir.
So just to let you know कि when the parasite gets into our blood and then it goes into our liver, okay, so there are no initial symptoms of malaria for about 8 to 10 days. पहले 8 से 10 दिन तक उसका कोई symptoms नहीं रहेगा. ठीक है? So this period is considered as something called as the incubation period or latent period. ओके, तो ये 7 से 8 दिनों में क्या होने वाला है कि वंस दे गेट इनटू द हिपेटोसाइट्स, दे द ट्रांसफॉर्मेशन ऑफ स्पोरोजोइट टेक्स प्लेस ड्यूरिंग दिस पीरियड. Okay, so what exactly happens is that it becomes spherical and is now and is now called as cryptozoite. So the very first morphological changes that happens in the liver hepatocyte. Okay, in these liver parenchymatous cells, the first transformation occurs from sporozoite to something called as cryptozoite. Okay, the sickle shaped parasite now becomes spherical. Okay, and at this stage, once it becomes spherical in nature, we no longer call it as sporozoite, we call it as cryptozoite. Okay, then further what happens is that there is considerable increase in the size of this cryptozoite. Okay, so initially when the initial cryptozoites are formed, they are about 10 micrometer in diameter. Okay, but as they grow in size, they can reach up to 40 micrometer in size. Matlab they increase about 4 times as the original size of the original cryptozoite. Okay, and this happen as the result of absorption of glycogen, fat and cytoplasm from the hepatic cells. Okay, now one might also think, okay, कि why this parasite might have chose to you know select hepatocyte. ये किडनी में जाके इन्फेक्शन क्यों नहीं करता है, लीवर में ही क्यों? Okay, so why do you think these parasite might have selected the liver cells than any other organelle from human body? Any idea?
So maybe liver acts as one of the checkpoints for metabolism. So
Yes,
maybe that.
yes.
Uh,
देखो, the thing is,
maybe. Yeah.
हाँ, बोला?
So because maybe liver, like know that liver tissues can self renew them for like compared to other organs, liver can self renew themselves. So because of that.
yes, it has got its pluripotency nature. It can regenerate itself if there is damage. But you should also remember that liver is the place where excess of the food nutrients is converted into glycogen and fat and stored for the
Mm.
reserve, right? If there is starvation, okay,
Mm.
the energy that is stored in form of glycogen fat, that is first occupied, that is first utilized by body. Okay, so just think the beauty of nature, that this parasite have evolved, right? They have selected the perfect spot where there is excess of nutrients already available. बरोबर है?
Yes, sir. Yeah.
Okay, so that is one of the reason that is said ki, okay, that this parasite upon entry into human body, it prefers to go and get colonized into liver rather than some another organism or some another organelle. Okay, okay, this is the reason. Okay, now let us come to the point. Okay, so now the cryptozoid so form, it has increased now 4 times the original size, right? And when it is increased in 4 times of its size, now we call it as schizont, or some people also call it as miront. In some textbook, you would see it that people referring it to schizont, while in some textbook people has referred it to miront. Okay, so again it enlarges and its nucleus divides by repeated mitosis into numerous nuclei like this. Okay, तो अभी ये बड़ा हो गया है, खा पी के टुल मस्त में मोटा हो गया, and then it has started its division now. The nucleus would now divide. Okay, and what kind of division happens over here? There is mitosis and a number of nuclei are formed. Okay, so now the cytoplasm is shared by each nucleus. Okay, तो ये हर एक न्यूक्लियस के आजू बाजू में क्या आएगा, थोड़ा सा cytoplasm आएगा. Okay, and a small bodies are formed in 1 micrometer in diameter, and these are called as cryptomerozoids. Okay, तो अभी क्या होगा, small bodies of nucleus, each and every nucleus would gather a little bit of cytoplasm around itself, and they would form small bodies called as cryptomerozoids. Cryptos stand for hidden. Since they are hidden inside the liver, those are called as cryptomerozoids. Okay,
Sir,
so बोलो बेटा.
all this process is still in the liver, right?
Yes, everything is happening in
Okay.
liver.
Okay. Yeah.
Okay, okay. Thus, from a single sporozoid that has infected, about 800 to thousands of cryptomerozoids are formed. Well, now do you see the amplification that a single parasitic cell has done? घुसते वक्त एक ही पैरासाइट अगर घुस गया तो पहले ही स्टेज में उसके कितने हो जाते हैं, 1 से 1000 के आसपास. ठीक है, some 800 to 1000 cryptomerozoids are produced by the process of asexual multiple fission. Okay, so now what happens next is that there are some authors that they have called this first division or first cycle of multiplication as a pre-erythrocytic cycle. Okay, ये क्या कहते हैं, we call it as pre-erythrocytic cycle. Okay, do you get this thing, pre-erythrocytic cycle? Okay, so now
Yeah.
whatever these cryptomerozoids are released, well, they have tendency to attack new liver cells. Okay, ठीक है. So okay, so now whatever these cryptomerozoids that are released from this already infected hepatocyte, when they are outside the cell, then of course they have chances to get entry into another hepatocyte. So this first phase of the division is sometimes referred to as pre-erythrocytic cycle or first phase of division. Okay, is it clear now? Is it clear? Shall I move forward?
Yes, sir.
Yes, sir.
Okay, okay. Now what happens next is that the cryptomerozoids which are liberated after schizogony, this process is called as schizogony. इसे क्या कहते हैं? Some people also called it as schizogony. The cycle of repeated division, asexual division, and release into its surrounding is called as schizogony. Okay. So these cryptozoids which are liberated after schizogony are capable of entering into either bloodstream or again attack the fresh hepatic cells. Okay. So now this cryptomerozoid which enters into the liver cell become they become either tropozoites. Okay, and these tropozoites are now known as metacryptozoids. Okay, तो ये सेकंड डिवीजन में जब ये फिर से दूसरे सेल को इन्फेक्ट करेंगे and when they get infected in second and permanent or you can say later kind of infective cycles, those are called as metacryptozoid. Okay, तो cryptomerozoite यानी क्या है? The product of first phase, that is pre-erythrocytic cycle, are sometimes termed as cryptomerozoid. But once this cryptomerozoid get out of cell and they infect another healthy blood cell, we now call them as metacryptozoids. Okay, उसे क्या कहते हैं अपन? We call them as metacryptozoids. And again, when they get entry into new liver cell, of course they would
Sir,
increase in size. Yes. Yes.
Sir, do they attack a new cell or do they attack a new? When they attack a new liver cell, then they're called metacryptozoid, or when they attack a new blood cell, they're called
No, no, no.
metacryptozoid?
We are not in blood cell. The story is yet to reach there. Okay. वो थोड़ा बाद में
हाँ,
आएगा.
okay,
We would
okay,
talk about that
yeah,
thing in
okay,
a bit later.
right,
अभी अपन सिर्फ लिवर में ही है. Okay.
okay.
So
Sure.
the subsequent phases
हाँ,
in which the metacryptozoid when get entry into new liver cell itself. Okay, दिख रहा है ग्रीन कलर का लिवर है. Okay,
Yeah.
and
हाँ, yeah.
in every subsequent stage of infection and multiplication, we would term them as metacryptozoids now. Okay. So
Okay.
now this metacryptozoid, when they
Okay.
get entry into new and healthy liver cell, they of course increase in size. Okay, and they are now called as schizonta or meron. Okay, then of course the nuclear division would occur. After nuclear division, they would acquire a cytoplasm around itself and the number would increase, the cell would burst. Okay, now ये जो cell bursting का यहाँ पर बताया गया है, well initially it was thought कि well it might happen due to shear pressure that is created inside the cell. Since the number of the parasite has tremendously increased inside the cell, this would impart very tremendous pressure on our liver cells and it would eventually get ruptured. That is what it was usually previously thought. But later on, well, people started to give some biochemical mechanisms that might be involved in release of these metacryptozoids out of liver cells. Okay, we would talk about that in details later sometime. Okay, in next few lectures as such, right? But for time being, you should be able to comprehend now once the nuclear division occurs, they collect a bit of cytoplasm around itself, they generate their own membrane and they burst out of cell. Okay, so in this way thousands of metacryptomerozoids are released. Okay, बात समझ में आ रही है आपको? Now these are called as metacryptomerozoids.
Yes.
Okay, so now the second phase of asexual multiplication, Okay, second and subsequent phases. Okay, what is released is something called as metacryptomerozoid. Okay, so now based on the morphology of these metacryptomerozoid, you can differentiate them into two types. Okay, इसे क्या कहते हैं अपन, मेगा मेटाक्रिप्टोमेरोजॉइड्स एंड माइक्रो मेटाक्रिप्टोमेरोजॉइड्स. ओके, तो ये जो मेटाक्रिप्टोमेरोजॉइड्स है ये दो टाइप के होते हैं, आप ऐसा कह सकते हैं. ओके, द बिगर इन साइज these mega metacryptomerozoids are bigger in size but smaller in number. Micro metacryptomerozoids are smaller in size and very much high in number. Okay, so this is the
Sir,
basic difference. Yes, huh?
I had a doubt.
Hmm?
So, like, if metacryptomerozoids, if they again attack new liver cells,
Hmm.
so
That is what they
like
do.
would they be called metacryptozoids or metacryptomerozoids? Because of
Metacryptomerozoids कहेंगे अपन उसे.
okay,
Okay,
okay, yeah.
okay, is it?
And
हाँ,
so
हाँ, बोलो बेटा.
one more question.
Huh?
Yeah, so this was Neha asked. So if these metacryptomerozoids attack new liver cells,
Hmm.
so again it will result in a subsequent cell burst and release,
Yes,
and that will again be metacryptomerozoids, right? I
yes,
mean,
yes,
there
yeah.
is no further development,
Further development.
or is
ठीक है. वेल देखो ये सारी चीजें जो है ना,
yeah,
मेटाक्रिप्टोमेरोजॉइड, मेटाक्रिप्टोजॉइड्स और क्रिप्टोमेरोजॉइड्स, वेल दे हैव बीन बायोकेमिकली कैरेक्टराइज्ड बेस्ड ऑन व्हाट टाइप ऑफ प्रोटीन्स आर एक्सप्रेस्ड ड्यूरिंग दिस सेल टाइप्स. मॉर्फोलॉजिकली अगर देखा जाए तो इफ यू लुक इनटू माइक्रोस्कोप यू वोंट बी एबल टू differentiate ki whether this is cryptomerozoid or whether this is metacryptozoid or whether it is metacryptomerozoid, right? Okay.
yeah.
So,
Thank you,
sir,
sir.
the cryptomerozoids, metacryptozoids, and metacryptomerozoids are essentially like very same in morphology. Is it
Very
the
same in
thing?
morphology, except this mega metacryptomerozoid and micro metacryptomerozoid can be
Yeah.
somehow compared on the basis of their sizes.
Okay. Okay. Yeah.
Okay, okay.
Hmm.
So now in this way, now can you imagine that this exoerythrocytic schizogony जो अपन बात कर रहे हैं, that is cyclic invasion and multiplication of this parasite. Well, in that would eventually act as a reservoir for this parasite. Okay.
Yes. Yeah.
Okay, and देखो, the thing is that when the parasite is present in this liver, well, the host's immune system cells cannot identify them, बरोबर है, because they are already present in host cell, बरोबर है, ठीक है.
Yeah.
And in this way it is one of the mechanism for this parasite to keep itself away from immune system, so that they remain hidden in the liver, safe from host immunity. Okay,
Okay.
but however, if in the case of any further immunocompromisation state, we'll say for example when viral load is present in blood, or we'll say for example if some virus infect the human being, the immunity then start to decrease, the immunity begin to fall, and in such
Yeah.
case whatever parasites which were present in this reservoir would come out and start something called as relapse. Okay, बात समझ में आ रही है आपको? तो ये जो लीवर के अंदर जो प्रेजेंट रहते हैं पैरासाइट्स, well those are the most problematic one, right? Because our immune
Yeah.
system के लिए those are inaccessible and drugs के लिए भी those are inaccessible. Okay, and
Oh.
these are the individuals that are responsible for something called as relapse. ओके, मलेरिया को ट्रीटमेंट करने के बाद में अगर पेशेंट ठीक हो जाता है तो कुछ दिनों बाद उसको रिलैप्स भी हो सकता है. तो ये रिलैप्स किसकी वजह से होता है? ये रिलैप्स होता है all the hidden parasite that were present in that liver cell. Okay.
Yes.
Okay.
Yeah.
So now this relapse का भी कुछ specific conditions है. And that usually is different according to species. Well, say for example, in case of Plasmodium vivax, relapse can occur repeatedly at least for 2 years. तो अगर ऐसा हुआ कि infection हुआ, okay, and infection has been cured by that patient, okay, and somehow in next 2 year he got infected by some virus wherein his immunity has been compromised, तो 2 साल के बाद में भी जो अंदर के पैरासाइट है वो बाहर निकल के आ सकते हैं. Okay, this is the
Okay.
case. In case of Plasmodium malaria, the period may extend to several years, right? On the other hand, if it is the case of Plasmodium falciparum, the relapse do not occur because the liver phases are self-terminating.
Okay.
इसका मतलब ये है कि Plasmodium falciparum ने अगर इन्फेक्शन हो गया, so it do not stay or remain hidden in liver. वो liver में से सीधा अगले फेज में चले जाएंगे जो मैं अभी आपको बताने वाला हूँ, that is erythrocytic schizophrenia और the blood cycle भी से. तो बात समझ में आ रही, ये रिलैप्स क्यों होता है, कैसे होता है?
Yeah. Huh.
Is it okay?
Yes, sir.
Okay. So heavy dose हो रहा क्या भाई लोग? Shall we stop? Shall we stop? Take a break and then come after some time. What do you say?
Okay.
Okay?
Okay, sir.
ठीक है. We will अभी क्या टाइम हुआ है then?
It's 4
4
o'clock.
बजे.
4 बजे है ना? It's 4 o'clock already. कब आते हैं फिर अपन, या फिर shall we stop now?
So next class वापस कब है? Next week को है क्या?
Next week को है.
Okay.
Okay, sir.
Shall we continue after a break or you think कि सर आज के लिए काफी हो गया इतना?
सर आज के लिए शायद
काफी? Okay. Okay, okay. कोई दिक्कत नहीं बेटा, कोई दिक्कत नहीं. Then maybe we would continue in our next lecture. Okay. ठीक है. Okay. So
sir,
do you know what
like-
your assignment is now for today?
Yes, sir.
Yes sir, वो पेपर पढ़ के उसका रिव्यू लिखना है ना, समरी टाइप, राइट?
Summary.
रिव्यू समरी.
Yes, yes, yes.
Okay.
Yeah. Sir, वो कब तक सबमिट करना है?
I will let you know the details.
Okay.
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 And sir, like, will you be sharing the presentation like after each topic is over?
Yes, yes. Of course, I would be sharing this presentation.
Okay. Thank you.
Okay.
Sir.
And whatever I have recorded, I will share that as well.
Okay,
Okay.
thank you.
Yeah, thank you, sir.
Okay.
Thank you sir.
ठीक है.